for the microbe project. From these seeds, we let them soak in buffer overnight, and then we take out all the microorganisms. These microorganisms will be then analyzed and preserved in liquid nitrogen. We do not preserve the seeds directly in the liquid nitrogen. They will not support that. We analyze them for the metabolism, what they can do with if they can metabolize sugar or not, etc. So this happens here. We have here an omnilog apparatus which permits to analyze 50 plates in the same time. So you have 25 trays like that. And these plates, in each well you have one particular metabolite, and you pour the microbiota on this, and there is some product inside which change color if the microorganisms together are able to use this specific metabolite. So we put the plates on this apparatus for 3 days, and in the back of the apparatus you've got a camera. We'll take pictures every 15 minutes, and when the microbiota can use the metabolites, the well turns purple, so you can see the purple color appears over time. And that's the way we do for analyzing the metabolism of the microbiota, which permits us to know what they can do and what they can't do. And that's permit to decide, to check whether the preservation of the microbiota has got an effect on the metabolism or not. The team's main lab, and for microbe here we store the seeds and we make the metabolic activities measurements. Molecular biology lab, so I'm not working today, so no lab coat. And here we perform all the DNA extractions and molecular analysis needed for the project. So here Cécile, who works at the CFBP with us, and she's in charge of the preservation of the strains and microbiota now. Yes. And here in this room, you've got the treasure of the collection. It means that all the strains are stored here in glass vials. So here at the collection itself. In each tray you have small cardboard boxes, and in these boxes you have the glass vials where we store the CFBP strains. Mm-hmm. So here you've got the CFBP strains. It was lyophilized in 2006, and here you've got the powder, which is the bacteria and cryoprotectants, and it's all lyophilized and stored under vacuum in this glass vial. Mm-hmm.
How long does this be stored, or forever?
We have vials which are 50 years old, mm-hmm. and we tried to revive them, and it works.
Oh, cool.
It's really, really efficient and really useful. Really easy, actually, to store bacteria. Well, these bacteria are aerobic. So we have here the 7,200 strains in the public collection, and we have other strains waiting to be available on the website. Okay, so that's the -80 freezer of the CFBP collection. So just a big -80 freezer, and that's it. It's pretty full right now, no problems. Here it's the lyophilizator of the collection. It's here we lyophilize the strains. And before lyophilization, the glass vials, they look, that's a glass vial before lyophilization with the strain. So we put a label inside, and then we sterilize everything. And after that we prepare the strain, we pour them at the bottom of the vial. And then we put a small cap here, a filter, and actually they are real cigarette filters. You know, these ones, the one we use to smoke cigarettes, and because they fit perfectly here, and everything is placed inside the lyophilizator. The lyophilizator will freeze overnight, then the temperature will rise slowly, and it will be all under vacuum. So when the temperature rises and goes over 0 degree, because it's under vacuum, the water will go directly from the solid phase to the gaseous phase. It's what we call sublimation. And the water will just be kept out, will be taken out from the sample. Everything will be dehydrated. After dehydration, after lyophilization, we take the vials out and we place them on this apparatus, which we call the hedgehog. In other collections they call that the Christmas tree. And we've got a vacuum pump here, so we make the vacuum once more. And then when it's all under vacuum, we take this heat sealing apparatus, and there is 3 flames here, and we can heat seal the vial. The flame will go there and the glass will melt, and because it's under vacuum, the side of the glass vials will just stick together and will close the vial. And that's because the bacteria are dehydrated and under vacuum. That will keep them alive for more than 50 years.
Mm-hmm.
It's really, really convenient because once they are on this format, you can send them anywhere in the world.
Mm-hmm.
And they can stay in the box for weeks and they won't be damaged. So that's the 3 fungus syncom used for microbe. And that's an example of what we can see on the plate when we cultivate them all together.
Just to show you a little bit of the result that I have for now, it's just a small graphic about the relative abundance of the genus, of the fungal genus. You can see here all of the condition. It's the same condition that we have tested here, the preservation condition. You can see every condition here. So it's a good representation to try to find a difference between this condition on the composition of the fungal community. So after that we also do statistical analysis to try to find if there is a significant difference between them.